Good afternoon all. I thank you all for the final Ph.D. presentation of Mr. Sonti. He is going to defend his thesis, and I thank all the respected members and managers who have joined us for this presentation. Now I request Sonti to present his data. Thank you.
Good afternoon, everyone. My name is Sonti Roy, and welcome to my presentation. So today is my Ph.D. Viva Voce, and I'll just proceed to the introduction. Yeah. So the disease that my thesis is associated with is called bovine theileriosis. It is also called tropical theileriosis, and it's a tick-borne disease caused by an epicomplexan parasite called Theileria annulata. And the symptoms of this disease include enlargement of lymph node, fever, then increased respiratory rate, labor breathing, anemia, and diarrhea. Now, the parasite belongs to a very important phylum called epicomplexan because it contains many other pathogens of human importance. And the tick vector that transmit this disease is of genus Hyloma. Now, in other species of Theileria, this may vary to other tick genus. Now, if you see the life cycle of this parasite, then it is completed in two hosts, the tick host and the bovine. Now, in the tick vector, it the sexual cycle takes place, and in case of bovine, the asexual cycle. Now, in the bovine, it starts with the inoculation of the sporozoites, and then it goes through various stages like schizont, merozoite, and pyroplasm. And the most pathogenic stage is considered to be the schizont stage. Now, my thesis is majorly focused on this stage of the parasite. And if we see the distribution of this parasite, you can see that it is found across India. And it is estimated that around $1.2 billion of loss occur due to this disease to the Indian livestock industry. Now, let's come to the pathogenesis. So, as I have told that the pathogenesis is due to the schizont stage of the parasite. So this stage have the capability to hijack the host cell and transform it into a cancerous type of cell where it manipulates various signaling pathway of the host and for its own advantage like cell growth. And there are certain hallmark of these infected cells like immortalization, hyperproliferation, and dissemination. So if we talk about the control measure for this parasite, then there are two methods. Like a drug is available, buparvaquinone, and then the Rakshavaxi vaccine. And this drug is only effective in the early stage of the vaccine, early stage of the disease. And the vaccine, it's a live attenuated vaccine. So and it have to be given yearly. Then because it's a live vaccine, so there is chances of reversal. Then it required cold storage, and it is country specific. So there is a fear that if we transport it to other country, then there may be introduction of new parasite genotype in that region. So it is never transported to other country. Then because it is generated through continuous passage, there is decrease in antigenic diversity, and which can have effect on the efficacy of the vaccine in the field condition. Now, this is one of the important point because my thesis also concerned with the genetic diversity of Theileria annulata. So when we look at the genetic diversity of Theileria annulata in India, there are no such study which talk about this, the diversity of the parasite as a whole in India. And if we understand the diversity, then it can help us to design effective disease control program and assess the vaccine efficacy in the field condition. Now, this vaccine have been used since multiple decades, but the attenuation mechanism of this vaccine is not known. So if you can understand the attenuation process or the virulence factor of the parasite, that how it is causing the virulence, we can identify new vaccine target or improve the current vaccine. Now, based on this is my study design where I'll be collecting samples from different region, and then positive samples will be taken forward for cell line establishment, which will be used to understand, do a dual transcriptome to understand this, the attenuation or virulence factor, to identify the virulence factor, and which involve both identification of important genes in host and parasite. Now we'll also assess the diversity of this parasite cell line. Okay. Okay. Yes. Thank you. So, and we have also done population genetics of the Theileria annulata, or population. We have understood the population structure, and where we assess the diversity, then genetic differentiation, linkage disequilibrium, and other parameters. So this is my first objective, where I was to establish the Theileria infected cell line, and this is the workflow where I have taken positive samples, and then I have gone through density gradient centrifugation, isolated the PBMC, and then cultured it RPMI and observed for sign of transformation. If it is transformed, then cryopreserved at early passage so that it can be used at the later study. Now we were able to it's majorly infect B cells and macrophages. So we were able to establish the Theileria infected cell lines, and this is confirmed using PCR and immunofluorescence, where we have generated antibodies to reconfirm that infection, and we were successful in establishing the cell line. Now let's come to the population genetics. So population genetics, before I talk about population genetics, I want to tell you what is population genetics. So population genetics is the study of genetic variation in the population and the evolutionary force that cause this variation. So evolutionary force can be genetic drift, mutation, or selection. Now why it is important to study population genetics? Because it will help us to understand the population structure, or is there any kind of selection, and can we implement some control measure which can control the diversity of the parasite. Now in case of Theileria annulata, the population structure of Theileria annulata in India is not known, and even the genetic diversity is also not studied at a pan level, and how the vaccine differ from the actual field population is also not known. And if we talk about the Indian parasite population, is there any selection pressure on the parasite population? So we can understand all those things through population genetics. Now the technique that I have, the marker that I have used for doing this, genotyping for the population genetics is called STR marker. It contains both micro and mini satellite marker. So it's a tandem repeats, and this repeats will be present in a region where it will be in a tandem manner, and the region that flanking this repeating region will contain the primer sequence. I mean, the primer should be designed in that region. Now there are 10 markers defined for Theileria annulata, which can be used for such study, and it is spread across the genome, which you can see that here the distribution have been given and the marker name also. And this marker have to be neutral in nature. Neutral means it should not be under any kind of selection pressure to give an unbiased population structure of that organism. And this marker were never used before this study in India. So this is the workflow that I followed for STR genotyping, where you can see I have taken sample, DNA from infected samples, and then PCR amplified the region, that locus. So the primer is flame labeled so that it's a fluorescent dye, so that it can be separated in capillary electrophoresis. And then this, after that, allele calling is done. That is how many bands are present or what allele is present. Then generating the genotype for each parasite or infection, and then doing the population genetic analysis. Now I want to mention here that when you do capillary electrophoresis, you get a chromatogram like this, and the peak represent a single band, and generally for a single locus it should contain single band, but here you can see that there are multiple bands. So it means that it contains multiple genotype of parasite. But for population genetics, we need to consider single allele for single infection, because we need to create multiple genotype. I mean mlg. Sorry. Okay. So you need to take the predominant peak. So predominant peak means just consider that a cattle is infected with 2 genotype of parasite and it contain 100 parasite. So 90 parasite, 90 parasite contain, have the same genotype. So we should consider the genotype of that 90 parasite, not the other parasite, which is of, 10 in number. So by considering that, we have constructed a MLG, which is multi-locus genotype. So single locus will contain single allele for that. And I will come into this, that what we do with other allele in the other slides. Now, in the population genetics, we have considered 4 regions in India, which is spanning across different geographical regions. And we have used STR marker to genotype the samples. And then we have performed this analysis to understand the genetic diversity and then population genetic differentiation, linkage disequilibrium, shared allele, and minimum marker. Now, yes. I didn't get it, sir. Okay. So RFLP, if I'm right, like RFLP is based on digestion, right? Restriction digestion. So because it's a tandem repeat and it can vary in length. So if you have certain sites inside the tandem repeat, this pattern will change, obviously, and you can do that. But mostly people use PCR and do the capillary electrophoresis. So your question is, will you get the same pattern? Okay. No, here multiple peaks means that when you amplify a single gene, you should get a single band, but you are getting multiple band, even though your primer is okay. And that means that you have multiple DNA in the samples, which have similar gene of different sites. So yes, but for population genetic analysis, all the standard tools consider single allele in a single locus. So we cannot do most of the analysis using multiple allele. So for some analysis, we have included single allele, but for some we have considered multiple allele.
Samples that you have obtained from different parts of the country, were these all cell lines or these are samples directly your genotype?
Yes, samples.
Samples.
Yes.
And even for the samples, within a particular state or an area, are they from a closely geographically closer places, or are they different? How are you actually selecting these samples?
Okay, so like in Telangana, I can say that I have also collected samples from many regions and then even collected from different collaborators. But in other state, it is mostly from collaborators, so we don't have much control over the region that we want to collect. But yeah, it is not so that it can reach every part of the state, but it is a mix of that.
I'm trying to think if you get a diversity. Is it because intra-host or is it inter-host?
Inter-host. Inter-host.
I'll let you see what
okay.
Yes. So it is specific for T analog, correct? No
yes. Yes. So first of all, genetic diversity, I'll come to the genetic diversity. So how do we measure genetic diversity? So there are 3 indices that I have used to measure the genetic diversity. One is called multiplicity of infection, then heterozygosity and rare allele frequency. So multiplicity of infection tell you about the number of genotype that is present in a single infection. And why it is important? Because it has been seen that higher the number of genotype, higher the infectivity or higher the pathogenesis. So if 2 cattle are there, one is infected with 100 genotype of parasite and the other is infected with 10 genotype of parasite, the cattle that is infected with 100 will have more symptoms and there is more chances that it will get infected. So
this is what I want to ask. When you say infected with 100 genotypes, is it because a handful of genotypes got into the animal and for certain reasons, in that particular animal, you got 100 different genotypes? Or did 100 different genotypes actually infect the animal? That's the question.
I didn't understand, sir.
When you say 100 genotypes,
Yes.
100 genotypes infected the animal. That means 100 different genotypes must have entered the animal at different times or same time.
Okay, so that we cannot predict that at what time, but when the animal is infected, it contains multiple genotypes because we have collected samples at that time. So
whether the evolution occurred within the animal or
yeah, and moreover, if there is a presence of multiple genotype in a region, there is always chance that this will do sexual reproduction in the tick, because tick will always feed on this cattle and then it will undergo, I mean, it can increase more diversity. So obviously the increase in diversity will happen in the tick vector because the sexual reproduction take place in the tick vector. But you are getting the genotype, because we are analyzing the samples from the cattle. So we are getting this date, I mean, genotype in the cattle. But yeah, I hope.
you must have some so across the different centers. Like you must have some inclusion criteria, like number of types of parasites.
Yeah, I mean, a perfect study will be where you can do a random sampling and then collect samples from every region and then every part of the region. But obviously, we need also samples which are actually infected with Theileria annulata, and then it may not be possible because like to collect from every part of every state. So yeah, there is this is one of the limitations because sample collection requires a lot of time, and then I would say that to do an unbiased analysis, I mean study, you need to collect it from or across India from every part of that, so that you get a very good population structure or how the genetic makeup is to understand that.
Do you also change the copy number of the
No. Total genotype we didn't see because we were interested in how the population structure is, and based on that we just genotype the parasite so that we can understand the population as a whole. And copy number, we didn't see copy number because this marker is already established. So it you are talking about the marker, right? So this marker should be present in a single locus. It should not be present in multiple locus. So it have to be present in a single locus so that
single clone of a parasite, when you analyze Sonti marker, you get a
yes, I mean here the data is not there, but when we do this in the cell line, we get very clear band, and that is single band. Even if you go to the capillary electrophoresis, you get very less band, but you cannot get, I mean, even the vaccine is not a single genotype, the parasite. So we do not have sample cell lines which are infected with a single genotype, but if you go to the cell line, then it reduces, but when it's a sample, we will get multiple bands because there is multiple genotype. And if you compare it, if you do the same cell lines, I mean, you will get the same pattern, I mean, in terms of the allele composition.
Doesn't this parasite also gather mutation while they are in the host?
Yeah, I mean, yes, mutation can, I mean, in the host, yeah, obviously there will be a mutation rate through which it can, I mean, acquire mutation in the, because it's a asexual phase in the tick, I mean, in the cattle, it can occur through, I mean.
You say that a sample, for example, a cell line, which is affected with your this thing, and then you sample it after a few more passages, you may see a genotype difference in them.
Because if I understand, like we are trying to analyze the length of the tandem repeats. So mostly we are concerned with if the length vary in size. So we are not looking at the nucleotide level that what changes is happening and is it different. But obviously this can occur because this is a natural process of, I mean, in every cells there is a mutation and every cell has different mutation rate. So mutation can occur, but we are not concerned with analyzing the mutation. We were just trying to genotype the parasite.
Tandem SPR length would differ, right? Like from point.
Yeah, I mean, if there is insertion, it will vary in length. But if we see, I mean, if you consider that, then it is not possible to use this as a I mean, for genotyping. There will be a random mutation and it will occur everywhere in the genome. By considering that, we think that, yeah, this is the length, and then it will vary in a very, I mean, in a tandem manner. Like if the length of the tandem repeat is 4 nucleotide, then it will increase by that length only.
So this primer, how many set of primers did you use for
10.
test? And did you evaluated this primer against the host that they do not find any, you know, they do not have
Similarity.
similarity with the host?
So
Did you test that?
this is a well-established marker, and there
You
is
did not design it? Did you
no.
use it from the published literature?
Yes. So multiplicity of infection, as I've told you, and then heterozygosity. So heterozygosity tell you about the number of heterozygote individual in a population, and then rare allele frequency. So rare allele frequency is the number of rare allele present in a population. So if I give an example, like why African population is considered to be the primitive and diverse, because the number of rare allele is very high in that population. So by that we can tell that that population is diverse. So we have used this, and we found that the Indian population has very high MOI compared to other countries till now reported, and even the vaccine, even though it is higher from other country, but in our population it is, I mean, from the Indian population it is lower. And then if we see the heterozygosity, it is comparable to other reports till now. And then in case of rare allele, what we found is that Gujarat have the highest number of rare allele. And based on that, we can say that Gujarat is having the highest diversity of population. And if we consider, then India have the highest number of, I mean, India have the genetic high genetic diversity of this parasite population. And even the MOI is also high in Indian population. Huh? Yes. So observe heterozygosity. So because this is a haploid individual, I mean parasite. So when you try to estimate the heterozygote individual, you need to know like which two alleles. Like if you can like if you consider the heterozygous individual, then both the alleles should be different. In case of diploid, right? It should have capital A, like what we have studied, capital N and small a, then it is called heterozygote. And if it is capital A, then it is homozygotes. But in this case, it is haploid. So how do you actually determine the heterozygosity? Because there are no heterozygotes. So it's actually haplotype diversity. And what you are asking is observe. So that the data that we are getting is allele frequency. And from that, we are extracting the observed allele frequency. And from there, we are calculating the heterozygosity, but it is not an actual heterozygosity. That's why it is termed as expected heterozygosity. I hope you understood. So because this is a count, and we are not comparing that. I mean, this is not that experiment that genotype in a single infection. In a single infection. Yes. Yes. Yes, because but the tools that it generate give a single value, and obviously it will vary, it will have a range, but here multiple locus are present, and ultimately you are averaging on that. So you cannot actually put a error bar. If you want to put error bar, you have to do it for particular locus, that what is the range it is coming for particular locus, what is the number of allele that it is coming, and it's a average of everything. So you can say that we cannot put it range over here.
Yeah, generally multiplicity of infection say that number of parasites infecting, number of. I don't know how much does starting your patient the parasite. So you based on the PCR thing, you are assuming that number of counts are there. How do you do?
Yes, I mean, look, the number of genotype in the infected cattle, we do not have any control over that. But when we isolate the samples, we try to estimate what is the number of genotype present in that samples. So how we do that? By,
I don't know how many cells are
yeah, I mean, even if cell doesn't matter, because we are trying to estimate the genotype, because and same genotype will give the same band. So obviously we are taking same amount of samples, enough samples, so that we can cover the whole parasitic load.
So this demo and you measure this, but it's actually given time, or it is a measurement of one period of time.
No, it is just a single point of time.
So you said that there is a vaccine is a mixture of genotypes. So do you see changing the genotype over a period of time?
That we have not observed, but obviously vaccine was also developed from clinical line, means isolated from infected cattle. But if we see the cell lines that is isolated from infected cattle have higher genotype compared to the vaccine. So through selection, it will obviously reduce the parasite that is more evolutionary fit will survive, and it will reduce to small number, or because it's a cell culture, is a selection, so it will reduce the number of genotype.
But since it's a cellular replication, it should stabilize it.
Yeah, I mean, in vaccine, it should be constant. It should be constant.
So this 5 is present for 5 genotypes? Or what?
Yes. So next comes the population genetic differentiation. So it talks about how two populations are different from each other. We want to understand that, and for that we have used three methods, that is DAPC analysis, then complete allelic clustering, and then K-mean clustering. So here you can see DAPC analysis, where the main objective of doing this is trying to see which population are different from each other. And what we found is that the three populations, that is Telangana, Haryana, and Gujarat, are similar compared to the Bihar population. And you can see this in the FST value also. FST value represent the genetic distance between two population. So here you can see wherever there is Bihar, it have a high FST value. So the genetic differentiation is low, but there are two genetic cluster if you consider the genetic differentiation. One contains Telangana, Haryana, and Gujarat, and the other is Bihar. Now next we proceeded to a random clustering method, where we don't assume any that there is any difference or similarity among the population. And by doing that, we try to cluster it, but we did not found so clear clustering in this method. So we next move to another clustering, where we are considering all the alleles. So what I have mentioned before, that we are considering only single allele. Here we are trying to take all the alleles, but you in the standard analysis tool, you cannot do that. So we have converted this allele into a binary format, where we have taken the allele. Suppose a allele is present in a sample, so we compare if it is present in the other sample or not. So if it is not present, we'll give 0, and if it is present, it will give 1. We did it for every locus, and we converted into a binary format and generated the genetic distance. And from there, we have generated this dendrogram, and you can see that these three populations, that is Gujarat, Haryana, and Telangana, are very close to each other. And while the Bihar population is far from that. So it tells you that there are two genetic cluster, but however, there is very low level of genetic differentiation among this population. Now we want to analyze the, we have analyzed the linkage disequilibrium. So linkage disequilibrium tells you about the selection pressure on the population. we want to see if certain genotype are multiplying more than the other. So in that case, what we found is that two population have a weak but significant LD, and this shows that there might be some kind of selection pressure going on this population. Now,
the animal population migration, or how this might be happening, looking at the Indian scenario of animal movement or whatever.
Yeah, I mean, so what I understand is that these three region might have movement of cattle, infected cattle is more compared to the Bihar, and that might is, I mean, in terms of genetics, we can say that the gene flow among this region is high compared to the Bihar, and that's how it have similar genotype in this region, and that's why they are clustering among themselves. And Bihar have little bit dissimilar genotype compared to these three region, and it might be because of less gene flow, and that can be because of no, we don't have no information. So next we turn to the vaccine, that how vaccine is different from the field population. So we try to understand the allele composition of vaccine and the field population, and we found that the vaccine share very low number of allele with the field population. So vaccine have been reported to work only against, I mean, it is effective against homogeneous population. Its efficacy is decreases in heterogeneous population. So it says that the vaccine need to be restructured because the field population is quite different from the vaccine population and in terms of allelic compositions. So we also analyze bottleneck. So bottleneck is a process where you try to see reduction in population size, and
Comments like that the vaccine work based on the available immunogenic protein. The allele systems that we have seen based on the tandem repeat is not a immunogenic portion of the parasite. So can you comment on that? Because you are not measuring here whether these changes in the or increase in the variation of the tandem repeat. If there is change in the immunogenic protein.
yes, but one thing is that we have taken a neutral marker which is not under any selection. So if two individual or two parasite are different in a population or have dissimilar genotype, then it should give different genotype or different profile. Yes, yes, not single means 10 markers we have used, and yes, heterogeneity. So obviously, if you want to actually genotype, you have to do whole genome sequencing and then try to understand what is the number of genotype in a population and compare it with the vaccine. Is it matching or is it different from the actual population? But obviously, cost comes, and then we have to genotype it using a marker which is feasible to do in this number of samples. So we have also done GBS for a small number of samples, but for this number of samples, we have used the SCR marker. So we try to see this is there any reduction, I mean, drastic reduction in the population size, and we did not found any drastic reduction in the population size. Next, we try to understand if the marker number can be reduced for future study. So what we found is that minimum of 4 marker could be used for this kind of study, and it will help us to reduce the cost and time of this kind of study, and can be implemented for monitoring purpose after implementing some eradication program. So we also try to estimate the diversity of the parasite, of certain parasite, using a TMS gene, and we found that even in that samples also but this number is low, the number of samples is low, and we found that it is also diverse in nature. So
more here. So among your four different regions, four groups, right?
okay. So no, these are not that comparison, because this is carried out on very small number of samples, and it is compared with previous data that were available across India as well as from other regions. For TMS, this data was available that people generally sequence that and try to estimate the diversity of the parasite.
From the four different regions of India.
No, they're not this sequence. We have not done that. So that's why it is not included in that, but we have tried to estimate the diversity in a small sample. Yes, these are the one which we have used, which are colored in red. So these are cell lines. We have used the cell lines to, I mean, we try to see the diversity of the cell lines. We have not used in the clinical sample. We want to understand is our clinical sample diverse enough, which I mentioned. Yes.
From certain regions.
Yes. So these are mostly in Telangana, because we need fresh samples for establishing cell lines. So mostly this contain Telangana samples. Yeah, it contain both B cell and macrophage.
That is the reason I asked. It starts from a single clone. What is the variation in there? Because any kind of protozoa might be infected with certain kind of either B cell preference or macrophage preference. We do not know at this point.
Yes.
Not analyze that way.
Yes.
It's a mixed culture.
It's a mixed culture, all right, and the number of genotype is always very high. So isolating a parasite genotype, I mean, it will be difficult, because in a single cells also there are multiple parasites, and it can be of different genotype. Yes, I mean, generally. Yes. People have done. So, like.
Is there any like group of that matter?
No, there is no such study that talk about that. And because if you want to actually infect the bovine cell, you need to get the sporozoid, and sporozoid is always present in the tick vector. So you need to get the sporozoid from the tick vector and then infect, because you cannot isolate the physont and infect again other cells, because that is non-infective. So I mean, there may be some method that could be developed, but right now there is no method like that.
What is the correlation between this antigenic diversity and your heterozygosity?
Heterozygosity and okay. So heterozygosity is the number of heterozygote individual in a population.
What Appa is trying to ask in terms of vaccine and vaccine failure, we are simply looking at allelic differences at places which do not have any bearing on the antigenic diversity. How do you actually hypothesize that these are failing because of that antigenic? Because that has no bearing on antigenic diversity. Unless that heterozygous loci are influencing it.
First of all, sir, like the STR marker that we use gives us a unbiased overview of the population structure. Yes. Okay.
Study is done.
Yes.
Heterozygosity in genotype and the
obviously. Yes, sir.
Antigenic protein.
Yes. Obviously, that need to be done to say actually the vaccine is not working in terms of that. But obviously, diversity, I think diversity is important for every disease, because if there is diversity in the actual population, I mean, if we talk about the COVID also, if there are multiple strain and the vaccine is made from its other, I mean, the single strain, then there is chances that it will fail. So we are just, yeah, yes, it is an extrapolation. Yes.
I can come from viruses. okay, even within the same individual, you will see what we call as quasi species. But the vaccine works. We can call the quasi species.
Okay.
Because there is a single serotype.
Yes.
A lot of variation is not required. You see a B cell epitope or T cell epitope. Again, that's right. Even though you see a lot of heterogeneity, vaccine may work or may not. That's the
Yes.
What you're claiming is might. You're not saying that this is not.
It might affect the efficacy.
So it is
So-
not possible to collect all population. Potential control.
I mean,
Even if you select.
yes, I mean, it should be possible. We have not done it, but what people have done is that they have taken a cell line and then they have infected the cell line with parasite, I mean, sporozoites. And but that also does not contain single genotype parasite. So, and mostly they were concerned with host, that what are the host changes. So they have taken a cell type like macrophage, a single type, or B cell, and then they have infected because they were not interested in the parasite genotype. So mostly there they are controlling the host cell type and not the parasite genotype, because obviously it will be tough to control, because once you get the sporozoite from the tick, it will have multiple genotypes, and to actually have a successful transformation, you need to inject every, I mean, all the sporozoites.
It will probably be and for it now having multiple genotypes. Do they have any difference in the compared to the that people are having less genotype in terms of multiplication or antigenetic?
I didn't understand.
So the genotypic differences are there or the
In host cell,
the patients are having multiple genotypes.
phenotypic difference. So, I mean, there is no such data that if the parasite vary, I mean, if certain cells have multiple genotype, what is the impact on host cells? So what is the difference in terms of phenotype? There are no such data.
Only genotype antigenetic except and something like that.
That is also not there. Yeah, I mean,
Like normal dilution, we could have separated these cell types, right? Actually, the host level. B cell, macrophage, right? It is possible, right?
yeah, it is possible. We could do that, but we cannot control the parasite genotype. That, yeah, host cell type can be controlled.
One more objective.
Yes. So the conclusion of this study is that we found high genetic variation among the parasite population in the country, and which suggests that they are successful spread in the country and could hamper the disease control program. Then this is the first data that is being like about the diversity and population structure of Theileria annulata in India. Then the low diversity in the vaccine might affect the I mean, vaccine efficacy, and then the reduced panel of marker identified in the study will be helpful in monitoring the parasite and its reintroduction after Theileria eradication. So these findings are the first report from India and would help in understanding the evolution and diversity of the parasite. Now next, let's come to the other objective, that is dual RNA-seq of Theileria lymphocytes. So as I've told that the vaccine is being available since decades, I mean multiple decades, but the attenuation process is not known. So can we understand the attenuation process or host parasite changes by comparing the attenuated line with the clinical line? We can, I mean, this will help us to understand even the hallmark that we see in the infected cells. And so our objective here is to understand the phenotype that we see in transformed cells, and then the transcriptional signature of these infected cells. In case of the parasite, we try to understand what are the genes that might be involved in virulent or pathways. So this is the study workflow where I have taken, I mean, infected samples and then established the cell lines, and then these are taken forward for RNA sequencing. Now we have analyzed for both Bos taurus and Theileria annulata, and different analysis were performed for both the species, as well as we also perform some combined analysis where we can identify both the processes that are enriched in both host and parasite. So this is the analysis pipeline that I followed. So here you can see I have taken RNA-seq-read, then quality control, and then mapping. So this is called sequential mapping, where because our cells, our data contain reads from two species, so we need to separate one species read before analyzing for other, and it is called sequential mapping, where you have to remove one species read before analyzing for other. And in most cases it should not cause problem if both the species belongs to different kingdom like bacteria and mammals, but in our case both are eukaryotes, so we have to separate that. And then we have performed co-analysis, co-expression analysis for both this host and parasite. So we got in case of host, we got around 2,800 genes to be differentially regulated and during Theileria infection. Now we want to understand how, what are the changes happening or which process these genes are involved in. But before proceeding into that, we want to confirm that is our data is actually in line with the previous results. So for that, we have selected previously reported 11 genes which were differentially regulated during Theileria infection and were already confirmed by other study using qPCR or Western blot. And what we found is that our gene expression patterns similarly match with there, and it confirm our data and verify the quality of RNA-seq. So we proceeded to other analysis, and we did KEGG enrichment, and what we found is that mostly cancer-related and other protozoan parasite-related processes are getting enriched in this gene list. Now, to reconfirm that, we went to ingenuity pathway analysis. So it's a software by Kaizen. Why it is why we have used this, because it contains database which are validated and from literature. So we have used that. And here also we found that cancer related signaling pathways are getting enriched in this gene list. And we also identify certain transcription factors which have this, which are dysregulated in our differentially expressed gene list and whose target are present in our list itself. So it can regulate the expression of those genes. Now, what we are finding is that mostly it is having this cancer-like signature. So we also this is one of the diagrams. Yeah.
So what is this? These are different.
Okay, okay, this one. Yes. Yes.
We are not able to see.
Yes.
What could be the reason?
What, sir? Can you repeat?
You can see the POS IDP.
Yes. 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 3 years. Yes.
What are these three?
So, like when you map it,
What is the down
BL03? Okay, this is uninfected, and these are infected.
So
all the TA are infected, and all the BL is uninfected.
So you think the inflammation is down
Inflammation is down regulating.
So I see a lot of things in IL-23 and all that.
Yes, we cannot. I cannot say specifically, but because there are multiple pathways that are getting affected due to this infection process, and this is the image where I have highlighted that there are multiple pathways which are getting affected, and several molecules are involved. In here you can see that there are many transcription factor which have this. Which have this molecule which are in our gene list. Zoom is not good. Can you repeat again?
I don't know what done but do you expect any change in RNA data when you do a comparison between infected which are having less phenotype versus more phenotype? Suppose any vaccine strain.
In terms of vaccine strain, we did, but obviously, we did not see it from that perspective. Like if we are having certain very low genotype number of, I mean, parasite and high genotype number, then what is the difference in terms of gene regulation? So we did not observe that. I mean, the study does not include that analysis.
So what changes you observed is that
So yeah, so here we are analyzing for the host because our data contain both host and parasite, and we have also used uninfected control. So we are trying to understand what are the changes that happen due to infection. So here we are trying to understand the process that is getting dysregulated due to infection. So we'll come later into that vaccine versus attenuate, I mean clinical. Yes, obviously it should play a role, but I mean it will be hard to design a study where you can control the genotype in a certain samples and then you will both the group. Yes. Yes, I mean we can do that, but it need to be passed very long period of time, certain cell lines, and the certain cell lines should have multiple, I mean at the initial stage of the development, like lower passage cell lines. Then only, I mean, we can control this parasite genotype. I mean, artificially, I don't know how to control that.
So these parasite genotype numbers go down over period of time?
Yes, because cell culture will obviously select certain genotype, parasite genotype, and slowly it will reduce. If you continuously passage for very 1 year or 2 year, it will reduce to lower number of genotype. But in case of if you collect samples from infected cattle, then it will have higher genotype. And at the start of the infection, like when you establish, at that time also it will have multiple genotypes, I mean large number of genotypes. So here you can see that we have mixed population. Yes.
So I have a question because when it is RNA, if you have to isolate up to different new population numbers, then you have variation. How would you account for different information in the house? And what I mean, then for that, 1,000 other parties of that, how they take an RNA sequence, how would you normalize the changes?
Yes, you are absolutely right. But one problem is that there are two way of thinking that should we consider the actual natural variation that is present in the field condition, or should we go with the laboratory where we just take single cell line and do a transcriptome study? Because in the nature we find because the parasite is able to infect two different type of cells, so obviously they have some similarities. So we want to find, and in case of RNA-seq, yeah, obviously if there is variation, then you will find a lot of genes. And I think we have tried to eliminate that because we are trying to observe that which is common in all those. I didn't get it. What?
I think that rather than doing the adaptation, the different population, you start doing more analysis of that, because that will also have some clinical impact.
Okay. So the answer would be that there are already studies which have done this kind of study. They have taken a cell line and they have infected with the parasite and they have done this transcriptome. But from the clinical point of view, we do not have data of a mixed population which is extracted from an actual infection. So we want to understand that what are the common processes in both the cell types that is getting actually upregulated or downregulated. Because in natural infection, it will infect both the cells. Obviously, in laboratory condition, if you get single type, then you will get less variation. You can easily identify which genes are getting dysregulated. But from a clinical point of view, it is important to understand the overall process. So taking into account that, we have considered both the cell line types.
Many of the time macrophages will adapt. So it depends on what time you are infecting yourself. And then if you take population like floating cells in the
Yes. Yes, I mean generally what Yes.
Majority of like you can root out other genes. Macrophages will have.
Yes. So when you establish the cell line, so generally those cells only will be able to multiply which have this infection, and we try to, obviously we should not take, I mean, if we don't want any kind of selection, we should take, I mean, both the cells which are adhered and even floating. So so next we proceeded to, does this theileria infected cell have any kind of signature, cancer-like signature? For that we have taken 34 human cancer cell line signature which are present in EBI cancer cell line atlas and compared to our theileria infected cell, with the goal of determining if theileria infected cells have certain transcriptional signature similar to any cancer types. So what we found that it is clustering with some B cell lymphoma, and it have similar cancer transcriptional signature, I mean, similar to this cell types. So next we extracted this gene expression data, and then we try to correlate the expression value of this cell line of all the genes with the theileria infected cell, and we found that there is high correlation among this expression pattern in both these cell types. And if we see this, these are the genes which are highlighted, I mean labeled. These are mostly reported to be involved in cancer. So we have highlighted them, and we found that most of them, many are present in our, I mean, comparison. And these are some of the representation of those genes which are present. So now the question is our research of host transcriptome an improvement upon the state of knowledge already exists in this area? So we want to understand what we are adding, like what we are trying to understand, is it new or is it already present in previously published? So for that we took a data which is published in 2020, but they have used laboratory established cell lines, and we try to compare them, and then we found that our data set contained around 2,138 unique genes, and we have already done enrichment of our data, and we found that these are mostly enriched in cancer-like signaling pathways. So this pathways, this is the conclusion of this host transcriptome, where we have separated all the genes in according to the signaling pathway, and these are mostly cancer related, and we have highlighted, if it is reported, we have highlighted in red, and all the black color genes are not reported till now. Next, we want to understand that what are the process in both host and parasite are getting enriched upon infection. So we want to understand in a combined way. So like, what is the process that is getting enriched in both host and parasite are using to fight the infection, and in case of parasite for their survival. So what we found is that 3 processes are getting enriched in both host and parasite, and these are splicing, proteolysis, and translation. Next, we proceeded to alternative splicing analysis, because alternative splicing have been shown to be involved in cancer, and it can lead to, I mean, defective protein formation, isoform production, and then it can lead to various progression of cancer and all. So for this, I mean, in this analysis, we have found that around 272 genes are being alternatively spliced, and some of the genes are already reported in cancer to be involved in, promoting the cancer. So these are the genes, and here I have highlighted for representation purpose one gene. Now we move to the parasite study, where we want to understand if can we identify parasite genes associated with theileria-induced pathogenesis using the RNA-seq data. So for that, we have divided the samples into 2 groups, attenuated and clinical cell line, and then this separation was confirmed using PCA plot, where you can see these 2 groups are clustered differently. And then when we performed differential analysis, what we found is that 71 genes were being differentially expressed, and when we classify them according to the family, we found that multi-gene families are being differentially expressed. Now we want to validate that, and for validation, what we did, the study that I have mentioned previously, we have extracted data from their data, and then we have done the analysis, and we found that these are the genes which are common in both the study, and it also contains this multi-gene family. So we can say that this, in case of virulent, there might be some role of this multi-gene family. So why it is important, the multi-gene family? Because multi-gene family have been shown to be involved in various like immune evasion and then immune evasion process in case of Plasmodium falciparum and then Trypanosoma cruzi, and this may play a significant role in theory of parasite virulence. So this is the conclusion of the parasite data, and overall what we conclude is that here we have used a combined RNA-seq strategy to better comprehend host parasite interaction. Yes. The higher passage and the vaccine cell line is the attenuated cell line, and yes, and which the cell line that is clustering with the vaccine cell line are being used as attenuated cell line. Yes. Yes, so that's why we have used the other study data to validate our data, because obviously we do not have a phenotypic grouping in case of this cell line. That are we finding, because in their study they have created this laboratory established cell line, and they have knocked down, they have created an attenuated cell line, and then they have certain cell lines which they are calling it virulent, and then they are comparing in that, and we have used their data to validate our data. Yes, yes, whatever it is coming in common, we are considering that this gene might be involved in the virulence. We are not concluding from our data itself, but we have compared. Yes.
Their rate of multiplication plus when you add growth and culture, or you add to cells or matrices and the rate of infection, you can categorize into different ways.
Rate of growth.
Yeah, I mean, how do you define highly virulent and
less virulent? Yes, so the other group that they are concerned with mostly host, but they define virulence as the property that the infected cell have, the host cell have, that is dissemination. So it should be highly motile, and it should be able to move. Yes, we could have done that. Yes. Infectious yield. Dissemination. Yes, I mean, in terms of that, matricial assay can be done where the cell actually migrate at a faster rate, and if it is faster, then you can call it that. Yes. So yeah, this is the conclusion. So our finding expand our understanding how novel signaling pathway and gene contribute to infection, and furthermore we demonstrated differential expression of unique host gene that might play key role in many cancer and parasitic disorder. Then we have identified certain genes through alternate splicing, and we have also identified multi-gene family that might be involved in parasite virulence. Importantly, this is the first study of filaria infected cells that have demonstrated to exhibit differential expression of multi-gene family. So previously, the multi-gene family have been bioinformatically linked to virulence, but there were no such experimental data. So our results provide a foundation for future study of how signaling dysregulation and parasite virulence contribute to theliosis. So that's it, and these are my research contribution, and I would like to acknowledge all the committee members, external supervisor, and lab members, and NIAB, DBT, and Manipal. Thank you. Yeah, please. Starter peak formation. Okay, yes. So if I come to the plot, 1 second. So what you're saying is that when there are very close peaks, so you need to, how you consider actually this is a single peak or multiple peak. So the tools that I have used have this filtering, so starter peak, and then how when you consider this single peak or multiple peak. So I have taken into account, and then by using some threshold, I have calculate. I mean, I have generated the allele counts. Yes, and yes, that we consider that, and must one more thing is that there are like two type of mutation model that one can happen that where the tandem repeat is being increased. That is, if the tandem repeat is just 5, then it will, the length will increase by 5 nucleotide every time, but somewhere it may increase by 1 nucleotide also. So there are two models of mutation that can happen, I mean changes that can happen in this kind of markers. So we need to consider, somewhere we need to also consider this single nucleotide or 2 nucleotide changes. So but generally I have, wherever there is chances of, I mean like single peak being split into 2, I have taken into account that. Yeah, good question, because when I have been establishing the cell line, so I have screened a lot of samples, I mean and then we try to culture them. So we don't know why certain parasite are able to establish itself in the cell and certain parasite are not. So this is a random process, and very low percentage of infection are actually getting converted into cell lines. Some cell lines were there where we have freezed, it is around maybe 20-30, where the cells never grow or never die out. The cell number remain constant and it is positive for Theileria infected, I mean Theileria infection. So we don't understand the actual transformation process, that why certain parasite are actually able to transform the host cell and establish itself. But that required, I mean, different study to understand that why parasite might do this. Yeah.
So which parameter or algorithm would you have set that usually identify both this locus?
No, this is just a formula. So like heterozygous, it's very simple. Just one, you subtract the homozygosity from 1, you will get the heterozygosity, and homozygosity you can calculate it from the allele frequency, you get it from genotyping. Like if you consider that if you consider A as capital A as allele, yeah.
Which algorithm that we have taken for the, please, I know the calculation, but for single locus we are using different algorithms.
No. This is for understanding purpose. We have multiple locus. We have used 10 markers, so that means 10 locus we have used. And this is for understanding that when you have single locus, you will just subtract the homozygosity from 1. And when it is multiple locus, you have to consider all the locus. So it becomes 1 minus 1 by m summation of 1 to m of homozygosity square. I mean that allele square. So yeah, normal representation, and in my case this applies for multiple locus.
What is the cutoff that you have taken for further, like for me, even you have taken a BAT, that is different component of principal value, right? Then the primal value and the BIC that must be validated before following the clustering pattern. And how much LD you have taken, because you have explained about the linkage between it. and you're looking at a different breed, and you are saying that you have properly clustered. So in that terms, I'm asking.
Yeah.
So the pattern very different. So which algorithm top of it in that manner? And again, if you look about it hypothetically also, I want to know about the LD cutoff that you are taking first. Because Haryana increasingly in the different slides is coming into different directions, right?
Okay, so you are talking about discriminatory analysis. So the main objective of DAPC analysis is to differentiate the group. Okay, so here what we do in PCA, we try to see the maximum variation into, I mean, where there is maximum variation in which direction. Here we try to maximize that variation for the groups, and by doing that we try to see what is even the slightest variation is there among those groups. So here what we found is that this, if we consider these two clusters, then there is very low level of variation. And what you are talking about the cutoff, then if I come into here, I have done that the value that you are talking about, but here it is not there, and I should have put it, but so one more thing, if you go to the actual paper of this method, you can see that they are themselves, they are mentioning that this is very random process and it does not assume any model. So while analyzing this result, you have to take into consider many things. So you can read the actual paper, I'll forward it to you, and there you can see that even though you have a high FST value, you may not get cluster in this graph. Yes, that's what I'm saying. The actual paper, you can read that, and there it is written that if you, even though you have high FST, you may not get cluster in this graph. So that is a little complicated. You have to understand the model, and then only you can understand it. Look, the tool that I have used applies that model. So obviously that the paper, that the method have been developed, I mean the tool that use the same method that is published in that paper. So yes, so I think I'll forward it to you. You can read, there is a specific line that if even though if you have high FST value, you may not get cluster in the clustering.
Like when you're taking population, you have to first of all filter them. You have to see them for the homozygosity when you're taking. You have to remove them, then only that is the name that is. And here it is not true. That is not true.
Yes, I have not shown that. So I'll show that. Thank you.
Okay, I know one question. So you said you put a transformation. So at what stage is integrated? So it is the most process, or is there any parasite protein kind of making the leukocyte transfer? Or is it possible to insert that particular gene from parasite into the whole and get the similar phenotype?
Yes, actually, that's what we try to understand. I mean, not the transformation process, obviously, but there is a paper from in Nature called TAPIN. I mean, TAPIN, they have somehow correlated with the transformation, but still, I would say that I mean, we are not addressing that. And moreover, there are some bioinformatics paper where they have predicted that there is a group of, gene family called TASH, and they think that the transformation process is conserved across Theileria genus. I mean, not genus, but two species, that is Theileria parva and Theileria annulata, have this property of transforming. So the gene responsible for transformation should be present in both these species. So by doing many analysis, they come to the conclusion that one group of family have this property of transforming. Like, it should be transported to the host nucleus to control the gene expression. It should be conserved in these two Theileria species, and there are many other criteria. And based on that, they have defined that this gene family might have the capability to cause this transformation. But till now, no experimental data is there that if we put this gene, then will there be transformation, because there are multiple genes in that family. And I think Yes, actually, I mean, locus means there are multiple genes. Yes. Yeah, it can be done. I mean, to test that hypothesis that if it is actually causing the transformation. Okay. Thank you.
So, just, external, you can see this on the-
Yeah, thanks, Sonti, for wonderful work. So, at this juncture, just I would like to congratulate you.
Thank you, sir.
Successfully defending your.
Thank you. Thank you, sir.
So now we can call you as a doctor.
Yes.
It's very good for you, okay?
Yes, yes. I'll-
Congratulations, Sonti,
Yes, thank you.
and welcome to the club. I hope you do well.
Yes.
Congratulations.
Thank you.
Yes, congratulations.
Thank you.
You are the first PhD student of Dr. Paresh. So congratulations to Dr. Paresh for getting his first PhD done. Best of luck with all your future endeavors. So where are you right now?
So I'm working at a company called Christian Hansen. So there I got a position called research scientist. So basically my work is in automation. Thank you.
If no more questions, I would like to thank all of you for coming for the presentation. Thank you to the respective members. Now we can join for some snacks outside, and I congratulate Mr. to Dr. Sonti Roy.
Thank you.
Congratulations.
Thank you, sir.